last couple of days. For those who are just arriving last night, it was patients and families only on a Sunday night dinner. And one of our patients, Ted Meyer, gave a wonderful presentation. And then on yesterday, on Monday, we had wonderful workshops all day. And these workshops were taught or facilitated by patients and family members for patients and family members. So they were small, they were personable, and there was lots of good information. We have an incredibly tight schedule today, so we're going to try as much as possible to stick to schedule. We're starting with a medical panel of 3 super rock star medical experts who give us so much expertise and love and care that in words, I know I personally could never show my appreciation. And so we have, I have to pull up my bios, we have Dr. Pramod Mistry, Dr. Sidransky, and Dr. Ozlem Goker-Alpan. And I'm just going to read a little bio about Dr. Mistry, and then Aviva will read with the other 2. Dr. Mistry, and I think everybody knows him, is the professor of medicine of digestive diseases and pediatrics, professor of cellular and molecular physiology, director of Yale Lysosomal Disease Center and Gaucher Disease Treatment Center. Thank you, Dr. Mistry, for being here, and I'll let Aviva do the other 2.
Good morning, everyone. And these 3 probably need no introductions, but just, to be official. Dr. Ellen Sidransky is the Chief of Molecular Neurogenetics Section of NIH. She is a pediatrician and clinical geneticist in the Medical Genetics Branch of the National Human Genome Research Institute at the NIH. Over the past 2 decades, Dr. Sidransky and her team have made significant contributions to the field of neurology with the discovery of the relationship between GD and Parkinson's disease. And Dr. Ozlem Goker-Alpan is the founder and CEO of the Lysosomal Disease Research Treatment Center, or LDRTC. She founded it in 2013 with a vision to provide high-quality care for individuals with lysosomal storage disorders and other rare diseases by utilizing a variety of clinical treatment protocols, multi-specialty clinics, and research protocols. So thank you all, to you 3, for being here, and we look really, really excited to hearing your talks. I'll sit there and then just. We won't be able to see if you sit up there. Oh, okay. Whatever.
Good morning, everybody. I want to especially thank Aviva and Cindy. I know that they have had a national impact through their advocacy and passion, and international impact because Aviva is on the executive board of the International Gaucher Alliance. And, Gaucher Community Alliance is something that truly resonates. This year is my 30th year being a member of the Gaucher community. And as many of you know, I have helped build programs across multiple continents, and it has been just an enormous privilege for me to share in your struggles, in your joys, and incredible accomplishments. And so the Gaucher community is indeed my family, as well as all of you gathered here. So if I can have my first slide, and then I would like, So this is a really historic meeting, I think. And I'm glad that you are taking videos so people who cannot come here can listen to the discussions by my colleagues, Dr. Sidransky and Alpan. So what we do is take care of the Gaucher community in 21st century. And everything for us begins with the patient. And every question that we have had, that we've taken to the laboratory, have started with a conversation, just like I was talking to a lady earlier on today. And what we do is our care is very focused on the pathway that the disease have followed in the individual patient, and then we apply the highest most sophisticated technologies in the world, including mouse models, including stem cell technology, genome sequencing, immune profiling, and biomarker discovery, so that I want to be able to feel at the end of the day that I'm actually helping every result, data points that we get is enhancing care of our patients. So Cindy and Aviva asked me to talk about inflammation, okay? And inflammation is actually a very historic concept, and it was first written in the ancient times. And a Roman writer, Cornelius Celsius, in the 1st century, described the first 4 cardinal signs of inflammation. And Rudolf Virchow, a celebrated pathologist, described the 5th one. And this was written in Italian, and so I will not make a fool of myself by speaking in Italian exactly what they wrote, but calor is heat. Rubor is redness, you know, when you have your bone crisis. Tumor is swelling, and we see that kind of swelling. And dolor is pain, and we now know also the loss of function that all of you are very familiar with. And in 1852, this is around the time Dr. Gaucher first described his case, 30 years later, Virchow proposed that when there is inflammation, there is cancer as well. And so I think that this is a very good starting point because this essentially describes what patients experience in their lives, okay? So I'm going to be describing, in your talk yesterday, you talked about patient narratives. And I'm going to frame my talk in terms of patient narratives. And so this is the first patient. I have this enormous privilege of taking care of Dr. Wayne Rosenfield, and he has given me permission to talk about him. When Wayne was 10 years old, he had a bone crisis in his right femur that was initially misdiagnosed as an infection of the bone. See, inflammation. Dr. Rosenfield describes this in his really highly acclaimed book, which has a rating of 4.6 over 5 on Amazon, Great Necessities, a Gaucher memoir. At age 15, he goes on to have right hip bone crisis. He was limping. And this was, children are teased mercilessly at school because of this, but also it was affecting his mobility, so he underwent osteotomy. In 1920, at the age of 25, he had infectious mono, okay? His spleen grew massively. See? Tumor in the inflammation. That's what happened in the spleen. It grew so huge that it ruptured, and had it not been for an emergency splenectomy, he would not have survived this crisis. So he underwent a hip replacement at age 35 that was revised after 30 years. And at age 40, he developed a very aggressive type of cancer, which has very poor survival, high-grade epithelioid hemangioma, and he had tumor resection followed by left above knee amputation. So this is inflammation, right? And so these are the kind of devastation. It's hard to imagine that one can function. And look at Dr. Rosenfield. He's a psychologist really making a huge impact on trauma victims and severe destruction of the vertebrae and his right femur. And he has had a second hip replacement. And this is since 2017. The previous one lasted 30 years. And this is the left hip that has undergone amputation. And you can see this huge amount of scarring, which is also embedded in the concept of inflammation. I mentioned that Wayne had huge spleen. This is not his picture, but this is a picture of a patient where the spleen normally is up here, is massively enlarged. And one surprising finding is that glycosphingolipids, the lipid, Gaucher lipid, is only 2% by weight, okay? And he had infectious mononucleosis. That's a virus that homes into B lymphocytes. And so findings like this immediately tell us that there is some crosstalk between Gaucher lipid-filled cells and the B lymphocytes. And so the 19th century was a very, very exciting time, and France and Europe in general was regarded as a center for medical innovation. And in 1882, when Dr. Philippe Gaucher looked after this 32-year-old woman who had massive spleen enlargement and liver and wasting, this lady sadly died at this hospital, Hospital Gaucher. And he did a very detailed study with pencil diagrams of what he saw under the microscope in the spleen. And he saw cells like this, and he thought that he had discovered a kind of a cancer, a new type of cancer in the spleen. At that time, of course, Dr. Gaucher thought that these were cancer cells and they were not macrophages, but this is Ilya Mechnikov, a Russian, probably the most consequential scientist ever to live, in my opinion. Ilya Mechnikov was regarded as an eccentric in Russia at University of Moscow. He did his PhD at the age 17. And he emigrated to France and worked at Pasteur Institute. Pasteur Institute is very close to Hospital Gaucher, by the way. And around this time, a few years later, Ilya Mechnikov discovered the macrophages, and he developed the foundational concepts about the role of inflammation that is still relevant today after more than a century. And we recognize immunity as something that is beneficial to us. You know, some of us have suffered from COVID, and we've recovered because of immunity. But in some people, that immunity can overshoot and become so hyperactive that it can lead to patients not surviving. And that is real pathological inflammation. And then what Ilya Mechnikov talked about is that inflammation is critical for maintaining harmony and disharmony, and that there is a physiological role for inflammation. So a controlled inflammation is beneficial, recovering from COVID. Uncontrolled inflammation is life-threatening, for example, deaths from COVID due to immune hyperactivation. So where does Gaucher fit in? Gaucher fits in right here, I would say. So of course, we now know that the immune system is very, very complex, and it involves making of antibodies from B lymphocytes, okay? And these are the antibodies that protect you from COVID infection, and they are generated by cells called B lymphocytes, but they are closely interconnected with T cells described here in a variety of T cells, but these macrophages play a very important role in controlling and initiating inflammation. So what do we know about Gaucher disease? Of course, we know Gaucher disease is a defect in the enzyme GBA. So one of the physiological role of inflammation of the macrophages in health is to process this lipid to break it down so that it does not build up enough for disease. But when this enzyme is low, it leads to the buildup of these lipids. And it turns out these lipids are very inflammatory. So we think about inflammation as something, a virus or a strep throat or a pneumonia, but you can also get inflammation from a chemical substance. And these chemical substances, which we now use as biomarkers, cause inflammation. Some people call it sterile inflammation because there's no infection. So Dr. Brady, and I think that at the first GCA meeting, we should definitely celebrate. Dr. Brady was the father of Gaucher disease. He won the Lasker Award, which is like a Nobel Prize. And without him, we would not be here today. But he and his group developed a program of research which was very macrophage-centric, because the macrophages are building up the lipids. And for a quarter of a century, they worked on delivering enzyme to the macrophage. And that has been very helpful because many of us are here today because of it. And the macrophages demonstrate this receptor which is used to target our enzyme replacement therapies into the macrophage and break down the lipid. But the macrophages also produce these substances that we call biomarkers, and the inflammation of the macrophage can be monitored very precisely by measuring this in individual patients in a longitudinal fashion. And this is a technical way of showing how precise these biomarkers are. And these are studies from my laboratory looking at Kaito, many of you know, CCL18, GPNMB. And if you put lyso-Gl1, it probably is the best property as a biomarker. So as I mentioned, this is a way to target enzyme. And this was demonstrated. This was, in fact, when I was a resident 30 years ago. This was the paper that really got me excited and wanting to belong to the Gaucher community. And what it shows is that enzyme replacement therapy, even if you put a tiny drop, it immediately goes to the liver, spleen, and the bone marrow, reversing disease manifestations. And all of these macrophage activation markers get better. And this was been shown with ERT, and it's been shown with SRT as well, as you can see here. So my story number 2 is about a macrophage and B-cell crosstalk. And this is Alan Gordon. This is a picture on the National Gaucher Association website. And I first met him in 1992 when I was a fellow with Professor Tim Cox in Cambridge. And he was diagnosed with Gaucher disease as well as smoldering multiple myeloma on a bone marrow biopsy because he had presented with a big spleen and low blood counts. And 3 years later, Alan passed away from myeloma. In 1990, he was active right to the end. He was very successful in business world. He was actually the founder, co-founder of the Gaucher Association, and he set up Alan Gordon Memorial Award and research grants, and I was the beneficiary of his generosity. But it was narratives like this that has made me feel that we really need to understand the biology and the science behind this to truly not have this kind of tragedies happen. So there are a lot of papers. In fact, one that just came out 2 weeks ago, an incredible paper led by Barry Rosenblum on cancers in Gaucher disease. But this is a study done by Tamar Taddy, who is professor at Yale. But when she was my fellow, she worked with me together with Greg Pastores to show that cancer risk was increased in Gaucher disease. Do you remember the first slide about Virchow, Rudolf Virchow? And then multiple myeloma risk was dramatically increased. And then there is a way of looking by this technique called Kaplan-Meier curve, and that is a way of saying what percentage of people are free of a particular event that we are looking at. So the antibodies I mentioned, immunoglobulins earlier on, there are many hundreds of thousands of different types of immunoglobulins, and we found that in total, the polyclonal gammopathy was very, very frequent in people with Gaucher disease that was followed by this pre-myeloma condition called MGUS, and ultimately by multiple myeloma. So I remember the day that Tamar presented this Kaplan-Meier curve to me, and I said to Tamar, the Gaucher cells are talking to B lymphocytes, okay? So this is an example of how very careful study of the trajectories in individual patient can give dramatic insights that can help us in turn to improve our ability to take care of patients. This is Shaini Nair. She is the director of my lab, and she too was inspired by this finding that you can have very big organs, enlarged organs, but the amount of lipid is of less than 2% by weight. And in a volley of papers, Shaini has described that when the Gaucher cells fill up with lipids, they present the Gaucher lipids together with molecules called CD1. And this is exactly how, let us say, COVID. When COVID gets into the macrophage breaks up COVID, and bits of, or even the vaccine, bits of the protein of the virus are presented, but they are different kind of molecules called HLA molecules. But these CD1 molecules are engineered to present lipid substances, right? And so when that is presented, then T lymphocytes come into action because they have receptors called T cell receptors, and these NK T cells that Shaini defined by very meticulous tests, then they stimulate B lymphocytes and they produce antibodies to the Gaucher lipids called lysogl-1 and glucosylceramide. So you see how parallel this pathway is between infection and inflammation in Gaucher disease. And this concept has recently been expanded further by work of Dr. Grabowski and others, where they've shown that these antigen-antibody complexes can activate the complement. And one of the effects of this complement activation is that they generate these molecules called C5a that bind to dock onto the receptors on macrophages, and the paradoxical effect is that it makes more of the Gaucher lipids. And so this is why treatment approaches that involve inhibition of this enzyme, popularly known as SRT, is a therapeutic target. So my story number 4 is this, and how the brain affects Gaucher disease in type 3 Gaucher disease. This is a young child, was on ERT, has been on ERT, is growing very nicely, but he has got subtle neurological signs, and he's 12 years old. And I show this picture with Richard's permission, and this is, sorry, I'm so sorry, Tad. And this is with his brother Richard that I looked after at Mount Sinai and followed him when he was a nursing home and sadly passed away, a gifted musician. Richard was a very profound sense of artistic beauty and so on that is also exemplified by Ted in his talk yesterday. So, so scientists as well as patients have generally thought of the brain as kind of separate. There's something going on in the brain, but it's not, other than thinking, there is no biological connection between the brain and the rest of the body. We are now learning that the gut microbiome can profoundly affect brain inflammation and brain function. Even sleep deprivation can affect brain function and brain inflammation. But there is this communication between the rest of the body and the brain on a continuous basis. And so for me, this has been the most profound revelation of our recent work. And you know, in the GCA program, I'm seeing that you've been talking about healing with art, therapy, and I think that all of these things actually do affect inflammation. Let me give you an example. Just this year, next week, a colleague of mine is presenting a paper at the American Liver Meeting where they looked at people with autoimmune hepatitis, and they found that meditation and mindfulness therapy actually improves autoimmune hepatitis. So the brain is in constant communication even with the gut microbiome and other organs in the body. And that's why I really appreciate the GCA's focus to talk about holistic care and not just the scientific biomarker monitoring. So when we come to the brain, there are immune cells, and when we are first born and conceived in the embryo, we think about the brain immune cells as coming from the bone marrow. That is absolutely not true, because in the brain, the macrophages called microglia come from the yolk sac, even on the second or third week of conception. And these microglia are in fact the first cells that go to the brain, even before neurons, and tile the brain, and then the neurons come in. And so these brain macrophages are derived from yolk sac. In contrast to the Gaucher cells that are pictures I showed you, they come from the bone marrow. This is very important to understand the biology of neuronopathic Gaucher disease. And so I was very pleased to see things like music therapy, how to live with chronic illness, relaxation, patient narrative through art, and having it all, and power of community and storytelling. I think that this is not all hocus pocus stuff. There is actually, we are going to find high-end science that actually impacts on the disease biology. So what we did was my team were able to design these spectacular mouse models where what we did was took mice with type 2 Gaucher disease, and then where the microglia don't have the enzyme, neurons don't have the enzyme, and these very important cell types called astrocytes. And so these cells are very important because for every neuron, there are 10 of these other cells like astrocytes and microglia. So actually, the brain has too many microglia and astrocytes and other cells than the neurons. And that in itself tells us that we really need to understand the behavior of these cell types when there is Gaucher disease. And so to do that, what we did was to rescue the Gaucher enzyme into individual cell types. And in the end, we had mice that had the Gaucher enzyme restored in the microglia, and also in mice that had Gaucher enzyme restored in the neurons. And then there was a very important cell line that we developed nearly 10 years ago, and these are healthy mice, but it's just a little bit of change, but it doesn't affect or cause Gaucher disease. And this technique of introducing this CRE allows us to only delete the enzyme in the microglia of the brain, okay? So this is spectacular model to say if the brain cells are not working, can we detect signals in blood to tell us what is going on in the brain? So as I mentioned, when you look at the immune cells in the brain, this is wild type, healthy mice. The main cell type is in fact the microglia. And when there is Gaucher disease, there is a reduction in the number of microglia, and all other immune cells are flooding into the brain. Then they are shown here, and these cells, all of these cells, are laden with Gaucher lipids. And so this is a technique called principal component analysis that helps us to see what is the nature of the healthy microglia and how do they differ from those microglia isolated from neuronopathic Gaucher disease. And it shows that they are very different type of cells. And so what we have learned is the microglia are like Dr. Jekyll and Dr. Hyde. So there are good microglia, Dr. Jekyll. These are very important. You know, in a single day, a microglia can be on the front of your brain and go back to the back of the brain many times, surveying the synaptic connections, hoping to form nerve pathways, and dealing with any danger, any kind of stress. And so the microglia are very, very important for doing synaptic pruning. It's a very big concept to form neuronal connection and regulation of neuronal circuits. And essentially, the microglia, without microglia, you cannot live. But there is a very Mr. Hyde-like property of microglia, and these microglia are found in neuronopathic Gaucher disease. And it turns out that these are exactly the kind of cells that drives Alzheimer's disease. They are been shown to be involved in Parkinson's disease, multiple sclerosis, ALS, Lou Gehrig's disease, as well as a type of cancer of the brain, and when you got infection in the brain, like viral encephalitis. And most certainly our work is showing that in Gaucher disease, neuronopathic Gaucher disease, it is involved. And this is a technique called single-cell RNA sequencing, and basically each dot represents a single cell. And this way, we open up the entire universe of the repertoire of immune cells in the brain, and it allows us to do sophisticated bioinformatic analysis to really understand what has gone awry with the microglia and how can we intervene in them in terms of treatment. And so we found that microglia were actually very, very important for maintaining Gaucher homeostasis, that microglia had the capacity to process nearly 85% of the lipids that are building up in the neuronopathic Gaucher disease. This is something I never anticipated, because until then, the focus of the work on how the neurons go awry, and clearly the neurons as well as microglia are talking to each other to cause the disease manifestation. This is a very busy slide. It is kind of pretty. I'm very proud of it. But I want to draw your attention to a biomarker that we found. We found that ApoE, I'm sure some of you have sent off saliva for Alzheimer's disease testing. I have, many other people do, for ApoE genotypes and so on. So ApoE is a serum protein that transfers cholesterol, and we found that it was dramatically increased, and after ERT it goes down, and the level of ApoE was related to glucosylsphingosine or lyso-Gb1 or lyso-GL1. and it is a very, very good biomarker. This is a pathway that we are making some pretty extraordinary findings that promises to really help us to look at neuronopathic Gaucher disease in a different way. So I mentioned to you, when we think about type 1 Gaucher disease trials, Dr. Brady would have never said to people that I'm going to recruit you to the trial because you have a bone crisis and expect that to get better. He had biomarkers like hemoglobin, platelet count, and large spleen. And that's how he was able to show the effect of enzyme replacement therapy. With neuronopathic Gaucher disease, our inclusion criteria is already having saccades or major squint or neurological signs not being able to balance. So it's like saying we are going to recruit people for a treatment just as if they have had a bone crisis, because those things don't reverse, and these neurological manifestations are just like that. And so one of our goals, a passionate goal I have had, is to see, can we identify blood biomarkers that may help us to identify earliest changes in the brain. And we found a marker, we believe, and in our mouse work, we found this neurofilament light, which is a product of damaged neurons that leaks out into blood, and it was elevated in our mice, and that correlated with the Gaucher lipid. Of course, we have a long way to go before we translate this to Gaucher disease patients, and we have seen promising trends in our patients. This neurofilament light is used as a marker for Alzheimer's disease, multiple sclerosis, Lou Gehrig's disease, and so on. So we are intensively working on this at the moment. So what we've learned is that when we reduce Gaucher lipids, in this case using the SRT that gets into the brain, we can actually reverse a lot of the immune activation involving the microglia, the NK cells, as well as the astrocytes. And this has been a really illuminating thing, that Gaucher lipids can drive this kind of immune changes, and we can reverse it by lowering the lipids, in this case using SRT. And so this is my last slide. I know Aviva is looking at her watch, forgive me. And so basically these, if you look at the crime scene, think about neuronopathic Gaucher disease as a crime scene. What do you find? You find this Mr. Hyde microglia, these macrophages that are coming out from the rest of the body, and these are NK cells that are working together to amplify the inflammation and ultimately promote progression of neuronal damage. And when this pathway can be significantly ameliorated if we restore the Gaucher enzyme in the microglia and reducing this inhibitor of GCS that can reverse this pathology even more. And at each of these stages, we've found biomarkers that help us to track what is going on in the whole of the brain. And so our data are in line with this paper that just came out 2 days ago that have shown in the phase 2 trial, LEAP trial, that there are significant effects of treating the brain in Gaucher disease in type 3. And there is now a clinical trial in progress. And so this is my very small group. You know, my research enterprise has been a mom and pop show. And this is the sole group that I have. But one thing unites us, which is what Winston Churchill said, never, never, never give up on our Gaucher community. And so with that, I thank you. And Aviva, if I went over my time, my
I think what we'll do is we'll have all the presentations. So Dr. Sidransky, if you could go next. And then we'll do a little panel of questions afterward.
Right, as they're getting my slides, hopefully, want to again thank also Aviva and Cindy. And I think, no, this was an enormous undertaking with lots of delays and contingencies, and it's been quite a feat. And I think it's just great to see Gaucher patients here together. And as somebody's remarked, and having been in the field for a long time, it's really remarkable to see how well the community is doing overall. The first meetings I went to, half the patients were in wheelchairs. This was really before enzyme replacement was widely available, and it's great to see you all. So I'm going to be talking about Gaucher disease and Parkinsonism. I'm actually a pediatrician. I never thought I'd be talking about Parkinson in many of the meetings I attend, but you go where the story takes you. So a lot of times when I give talks, I show this slide because I really like this quote. It says, "I live in a very small house, but my windows look out on a very large world." And my small house is the house of Gaucher disease, where I've been working for over 3 decades now. And you guys all know what Gaucher disease is. But from this house, we have a window into a seemingly unrelated complex disorder, Parkinson's disease. But I think I'm going to show you that our knowledge about Gaucher disease may actually advance the field of another disorder, and it will have a lot of benefits for us as a community also. So in our laboratory, we're using all kinds of different techniques in order to try to understand this link between the two disorders, which I'll be describing to you. But I think that by the end of this 20 minutes or however long I talk, you'll appreciate that disorders like Gaucher disease can really inform other diseases, and it's further justification of why America, the world, needs to support rare diseases. Not only do the people with rare diseases deserve the support, but by understanding rare diseases, we can also have insights that are applicable to others. So the questions I'm sure you're all thinking is isn't having Gaucher disease enough? Do I have to worry about another disorder, and it's not a very nice disorder, and what do I have to do? And so I want to try to give you some insight into it, but since I'm one of the first speakers, I just wanted to remind everybody that Gaucher disease is the inherited deficiency of the enzyme glucocerebrosidase. Sometimes I'll refer to it as GCA, and GBA is the gene that's mutated in Gaucher disease. And as you know, patients with Gaucher disease have very wide variation, and in fact, that's really what drew me to the field and what kept me engaged for all these years. It's divided, as you all know, into 3 types, but really I see it more as a spectrum where we have 80-year-olds who don't know they have it to babies that die in utero. And in the spectrum now, Parkinson manifestations is sort of falling among the things that we can see with Gaucher disease. And as you all know, Gaucher disease is a rare disease. Parkinson's disease, in contrast, is a common disorder. It affects about 2% of the population over age 65. There's about 40,000 new cases in the United States every year. It affects about 1 million people. And we know that the etiology of Parkinson's disease probably has both genetic and environmental components. Just to define it, the symptoms of Parkinson's disease, by definition, is bradykinesia, which is slow movements, and then at least one of the following: muscular rigidity, rest tremor, but a special kind of rest tremor, and postural instability. And this cartoon shows some of the other features that you might observe in a patient with Parkinson's disease. We also use the term Parkinsonism, and that's describing the motor features of Parkinson's disease. And I might also mention a related disorder called dementia with Lewy bodies, which is rarer than Parkinson's disease, but has some of the same features, but more severe cognitive deficits and a more rapid progression. Disorders that have Parkinsonism are also referred to as Lewy body diseases or synucleinopathies. So these Lewy bodies are these inclusions that you see within neurons, and they're made up of a bunch of different proteins, but among them is something called alpha-synuclein. And alpha-synuclein is this amorphous type of protein that tends to aggregate in clumps. And we see these Lewy bodies when we look in the brain of patients with Parkinson's disease. They kind of look like a target. And you'll hear a little bit more about that as we go. So for me, this whole story of Gaucher and Parkinsonism began for me with a single patient that I saw in the 1990s at the NIH Clinical Center. She had Gaucher disease, but she had also developed Parkinsonism. And her Gaucher disease was relatively mild. It was diagnosed at age 19. She had been splenectomized, but by the time she was 42, she had developed a tremor, rigidity, masked face, difficulty initiating movements, and a rapid deterioration of her gait. And we saw her at the NIH, I think about 3 times, a couple years apart, and we were upset to see that she had progressive dementia, and she died actually at age 54. So at first I thought it could be a coincidence. Just because you have a rare disease doesn't mean that you're immune from other common things. But when I started to talk to other Gaucher investigators around the world and dig through some really old literature, and also a paper that had been published that year from Israel and Italy, it turned out there were other cases of Gaucher disease with Parkinson. So we collaborated with centers around the world, and in 2003 we published a series of 17 patients with Gaucher disease who also had Parkinson's disease. And for me, the story might have ended there. I had my paper, and it was an observation. And then the plot sort of thickened. This is because a student that had worked in our lab a decade earlier gave me a call one day and said that she was now a pathologist in Boston at Mass General. And she was doing an autopsy of a patient with Parkinson's disease. She read in the chart that the patient had Gaucher disease, Googled it, found my paper, and said, do you want something from the autopsy? And of course, I was delighted to get a brain sample, but I said, I don't have any other samples from patients with Parkinson's disease. So can you send me a few? And so these 3 frozen brain samples arrived on dry ice. And it was really hard to read which one was which, so we decided to measure glucose cerebrosidase in the 3 brain samples. And actually, it was low in all 3. So I said, what the heck? Extract a little DNA from each of them. And one patient had 2 N370S alleles. That patient was the one with Gaucher disease, but the other 2 both carried Gaucher mutations. And this really blew me away, and I called this pathologist back, and I went to brain banks around the country. And within a few months, we had examined 57 different brain samples from patients with Parkinson's disease, and 12 of them had mutations in the Gaucher gene. The reviewers were skeptical, so we went back and tried to get controls from the same brain banks, and we didn't see any mutations in the controls. But I have to say, this study was very hard to get published. What's kind of ironic is the only people who weren't totally surprised by this was the Gaucher patient community, because some of the patients were telling me that when they would get together late at night after meetings, they would talk about their families, and they'd be complaining that they had parents or grandparents with Parkinson's disease. And in fact, we started doing a prospective study of all the patients that came to our clinic over about a year, and 12 out of 45 of them had a first-degree relative with either a parent or grandparent who had Gaucher disease, who had Parkinson's disease, who was a Gaucher carrier. And this was seen in other clinics like the Center in Jerusalem. So this was further evidence that heterozygotes or carriers were at risk for Parkinsonism. Again, even though different centers were finding this, there was still a lot of skepticism in the community. And so eventually, we were challenged to put together a multi-center study, which we coordinated and eventually recruited 16 Parkinson's centers around the world. Some of them are shown in the map. And they looked and did Gaucher genotyping on 5,000 patients with Parkinson and 5,000 controls. And we pretty much definitively showed that even screening for 2 Gaucher mutations, patients with Parkinson's disease were 5 times more likely to have a Gaucher mutation. So this pretty much sealed it. And even from this early study, there was some evidence that the mutation carriers might have had an earlier onset of Parkinson's disease and some more cognitive deficits. A few years later, we went back, and that other disorder that I mentioned, dementia with Lewy bodies, it's rarer, so it was harder to get as many cases, but it turns out that the odds ratio, the prevalence of Gaucher mutations in this group was even higher. And so it's clear that somehow mutations in GBA have some connection to these 2 neurodegenerative disorders. Well, why is it important? Well, finding a gene gives us a starting point. And from that, we can try to understand what the protein the gene codes for does. And in the case of Parkinson's disease, GBA kind of drew our attention to lysosomal dysfunction. And in fact, today, when you look at all the genes that have been associated with Parkinson's disease, many of them are falling into these lysosome pathways. Today, if you do a Google or a PubMed, which is how we find publications, and you put in the word GBA or glucocerebrosidase, you will find many more publications about Parkinson's disease than you will about Gaucher disease, so it's kind of ironic. This is when we first had the first paper, and now it's like 70, 80 a year. It's really hard for us all to keep up. And the other thing that's remarkable is the interest by pharma. Now almost all the big pharmaceutical companies, Merck, Lilly, Roche, Pfizer, they all have programs targeting glucocerebrosidase. So as I'll tell you a little more, I think that there is some good news for us from this all. So for now about 15 years, we've been studying patients to see if we can find early clinical or imaging features that are predictive of Parkinsonism in our patients with GBA mutations. And so Dr. Grizel Lopez, who's my neurologist, has coordinated a study where we recruit to the NIH patients with Gaucher disease who have Parkinson's disease, carriers of Gaucher disease with Parkinson's disease, and at the same time, we want patients that don't have Parkinson's disease and carriers, but do have a family history. And so when the patients come to NIH, and I recognize many of you in the audience that have been there, we do a physical and a neurologic examination. We do these annoying but interesting neurocognitive evaluations. We test their sense of smell, and we make you do all kinds of questionnaires for other non-motor symptoms. And then it's part of the time we're doing some imaging studies. We're still doing this ultrasound study of looking at the deep midbrain structures in the brain. So this slide is just a single slide, but it's summarizing like 15 years of work. So the patients have come, we have them seen by one neurologist and one neuropsychologist. For about 11 years, we were doing these PET studies, and we've had more than 500 visits. So what I'm summarizing is the patients with Gaucher disease or Gaucher carriers who have Parkinson's disease often resemble run-of-the-mill Parkinson's disease. Some of them have the features of dementia with Lewy bodies. They respond to Parkinson medications similarly to other patients with Parkinson's disease, but they tend to have an earlier onset of their Parkinson's disease, about between 2 and 6 years earlier. The mean age of our cohort was about 49 when they first developed Parkinson's disease, which is relatively young. They're more likely to have a positive family history of Parkinson's disease. They tend to have what we call non-motor symptoms and some problems with the sense of smell. They tend to progress faster, and they tend to have more cognitive issues. But this isn't composite. This is sort of when we look at all the patients that we've seen together that have both Gaucher disease and Parkinson's disease. When we look at individual patients, there are exceptions. Some of our patients have don't have this faster progression, and some of them have had no cognitive problems. So you can't always draw conclusions from the collective experience. The other thing that's been interesting is when we study our patients without Parkinson's disease, but even those that have a positive family history, it's been sort of frustrating. We've brought in about 93 individuals, 55 with Gaucher disease and 38 carriers. Some of them have been to us multiple times. And some of you are in the audience. And of all these patients, there's only one that we've followed for up to 13 years that developed Parkinson's disease. So it's clearly not the majority. And I think it doesn't bode for great publications, but it is very good news for the Gaucher community. One thing that we've been focusing on also are siblings, and we've recruited about 10 pairs of brothers or sisters with, you know who this is? It's an old TV show that we used to like in our house. But anyway, we had about 10, and the fellow on the left is Michael J. Fox. I didn't get his permission to show this, but he's all over the web anyway. He was the annoying older brother in this family. In any case, the siblings that we've identified all have Gaucher disease, but one sibling has Parkinson and one doesn't. And so we've brought them in, frequently, doing our complete battery of evaluations. And so far, none of the patients who didn't have Parkinson at the beginning have developed Parkinson in our group. But each time they come, we collect all kinds of biosamples and we do the gamut of tests, because I think that these siblings will be very important as we try to dissect out what protects you from Parkinson's disease and what predisposes you to Parkinson's disease. Because I do want to stress that most of our patients with Gaucher disease and most carriers don't develop Parkinson's disease. It's a risk factor, but it's not like Gaucher, where if you have 2 mutations, you're going to get Gaucher. If you have a Gaucher mutation, you're at increased risk, but might be 3 or 5 or 10 times more likely, but it's still a smaller number. So our challenges are to identify the factors or genes that either increase or decrease your risk for Parkinson's disease, both through these clinical evaluations, but now the time is ripe for doing different genomic analyses. Because today we have these great new techniques that enable us to unravel the factors that contribute to each of our individual tapestries. And we can look at DNA and RNA and proteins. And we want to see what's shared by the patients with Gaucher disease who develop Parkinson's disease. I do want to stress that collaboration is really important, that none of our centers have enough patients with Gaucher disease to do the mathematical calculations, but perhaps as we put centers together, it may be more possible. The other thing I want to mention, and it's a topic that doctors are not always that comfortable about, is tissue collection. One thing that's helped our studies measurably has been the ability to have some brain samples from patients. They're exceptionally valuable, and they're a rare source of material for all kinds of investigation. They're collected at autopsy, obviously. We're not going to take any chunks of anybody's brain. And autopsy is not something that everybody culturally or religiously is interested in doing. But if it is something that you're interested in, it is something that needs to be coordinated in order for the samples to be valuable. And sometimes patients end up having an autopsy for an unrelated reason, and it would be great for the Gaucher community to be able to get, the investigator community to be able to get access to some of the tissues. There are regional brain banks across the United States that will accept donations. Patients that have been to the NIH, sometimes we can arrange to coordinate an autopsy at NIH. But we think that comparing samples from patients with Gaucher disease, both those that develop Parkinson and those that don't, will help us to better understand what's going on. So the important question to us as a community is, can this Gaucher-Parkinson link lead to improved therapy, both for Parkinson's disease and for Gaucher disease? as we know, there are good drugs for Gaucher disease, but they're expensive, they're inconvenient, and they don't cross the blood-brain barrier. So there are strategies that you're going to hear about later today from different companies: gene therapy, substrate reduction, ways to get the enzyme better into the brain. What our group is focused on for about 15 years now has been the idea of chaperones. And this would be a disease-modifying therapy where you try to promote the activity of glucocerebrosidase by helping the mutant protein fold better to recover lysosomal function. Well, this is the sample that Cindy asked me to put, the photo that Cindy asked me to put in. This is a patient, Gaucher spleen. To do our screening, we actually used this sample from a patient, and collaborating with a new institute at NIH that does high throughput screening, we used these robots in order to take this mutant sample of glucocerebrosidase and screen, at the time, about 250,000 different compounds or chemicals to see which ones might affect your glucocerebrosidase activity. And we did identify a few. In fact, 2 of them were compounds that nobody had seen before that we thought would be interesting. And we worked on them a bit, and we actually showed that if these are Gaucher macrophages and they're loaded with the lipid in green, when we could treat the macrophages with one of these drugs, we were able to get rid of storage. We also made models of Gaucher disease by taking these fibroblast samples that patients have donated over the years and then reprogram them into cells that resemble, that are like neurons. And we found that what we call our iPSC dopaminergic neurons, these are the neurons that are affected in Parkinson disease, have decreased levels of our enzyme, and they store the lipids. So they they mimic what's seen in patients, so they're a nice model to work with. and this pretty figure, these are neurons, and you can see what's shown in green is where your glucocerebrosidase is, and what's shown in red is where the lysosomes are. And in controls, you see a lot of glucocerebrosidase in the lysosome. In some of our patient samples, don't see very much yellow because you don't get very much of the enzyme into the lysosome, but when you treat with one of our compounds, you see much more yellow, which is showing that some of it is getting delivered to the lysosome. We also, I mentioned this protein alpha-synuclein, which tends to be elevated in patients with Parkinson disease. And so in our cells from patients with Gaucher disease and Parkinson disease, you can see that this band is darker than the control band, which is indicating that we see more alpha-synuclein. And then when we treat, the levels seem to go down. So this is preliminary evidence that drugs that increase glucocerebrosidase may someday work to treat Parkinson disease. But clearly, there's lots and lots of work ahead. So my take-home messages today are that mutations in the Gaucher gene are a risk factor for Parkinson disease, even in carriers. However, most patients and most carriers will never develop Parkinson disease. I believe that learning more about this connection will improve our understanding of both disorders, and new drug development for Parkinson disease may ultimately be beneficial for Gaucher disease also. So how to contribute to this research? Well, you might want to get involved in a study. NIH, we've been studying Gaucher disease for quite a long time. I think we've got about 6 decades of samples. But there's other ways. Michael J. Fox Foundation is collecting patients with Gaucher disease to see if they can study Parkinson disease. And many of the other research centers, I'm sure you'll hear about, both from Ozlem and from Pram, are interested in seeing patients. Both patients with Gaucher disease and carriers who have Parkinson disease or don't have Parkinson disease are very valuable for this research. Consider sample and tissue donations. And keep up to date on new therapies. Go to clinicaltrials.gov and the websites and see what's going on in the field. So just to introduce our clinical team, Emery Ryan, our clinical coordinators here today. I mentioned Grisel Lopez and Nahi Taibi, who does a lot of the patient genotyping. Our emails are shown here. You can Google our names. The other thing is we are really interested in finding people who were at the NIH, even if it was 40 or 50 years ago, because we want to update our records, find out what's happened with you. And also, we need to update research consents, because what you consented to 40 years ago is not directly relevant to what we're doing today. We couldn't have dreamed that we'd be doing things like genome sequencing and iPSC cells. So I hope I've shown you that studies of our rare disease can come together and provide a really unique window into another disorder, but that also all these connections may someday come together to give us new therapies for both diseases. And I want to acknowledge, this is like a cast of thousands of people in my group, the people at NCATS and drug developing, Michael J. Fox, that's supporting a team that we work together with 5 investigators, collaborators both at NIH and around the country. But I always give special thanks to our patients, their family members and caregivers, and their referring physicians, because without them, this work is not possible. Thanks.
Hello, everyone. Actually, it is great to see the familiar faces and who are friends, and also we are welcoming new friends. I'm Ozlem Goker-Alpan. We actually are members of a very small, independent research and clinical care unit at Northern Virginia. And how our saga started actually with the shortage of actually Cerezyme. That is the day when I actually went out of NIH and started this clinical research unit at a townhouse, 2nd floor. The garbage wasn't collected. I just bought actually an infusion pump from an eBay, and we went and picked up another infusion chair from Long Island that we bought on the Craigslist, and the rest is history, okay? Yeah, we follow currently around 400, close to 400 patients. We started with Gaucher disease. In our repository, probably we collected 1,000 specimens for the past 10 years. Anyhow, so I am delighted to be one of the first speakers of this meeting, which is the inaugural meeting. Cindy called me one of the, I think, President's Day or whatever, we were in a ski trip, and it's like she said, oh, we are starting this new Gaucher group. Would you be interested, to work with us? I said, I'll be delighted. I'll do anything what you want. Okay, all right. So what she asked me to talk about is about type 3 and neuronopathic types. So it's mostly children, but we follow adults, and we have about 40 patients that we follow with neuronopathic Gaucher disease. I think we are one of the largest groups here. So I want to make this science-based, but it is going to be as practical as it gets, okay? The reason is, at the end of the day, very eloquent talks from Dr. Mistry and Dr. Ellen Sidransky, but we want to treat the patients. We want to find cures, and we want to have them to survive, and to survive well, okay? That is the aim of what we do. So I am going to do a prelude. I didn't want to put emphasis on this, because I am sure you have known this ad nauseum. I'm just going to give some pearls as we go along the slides, and what it means for a patient, when we look at the Gaucher cells, this and that. So basically, we start with the molecular change. This is what happens during conception, is that right? Either you inherit it from each parent, or there are some other events that leads to the molecular change. So basically, you have a mutation in the gene that's called the GBA1. It is officially now GBA1, and because there is a GBA2 too, and then it starts within the lysosome first. whereas the deficiency leads to inability to actually break down what we call is the substrate. Glucose is ceramide, it's a membrane, it's in the membrane, but what happens is basically the dysfunction spills from the lysosome to the cellular environment, and that goes to the tissue, and then the macrophages in the tissue, actually they either get engulfed actually in the periphery of the red blood cells, but in the tissues, they become different than macrophages. These are called Gaucher cells. They are huge, multinucleated. If you stain them with certain stains, they have a very specific appearance. These are actually activated macrophages. And they get activated for various reasons that I'm not going to go into detail. We can discuss in some upcoming, I think, sessions. They're both due to substrate, also the other inflammatory patterns. So we follow actual macrophage activation markers. This is chitotriosidase, which is commercially available, and CCL18 that is not available, but it's another marker that we do, and Dr. Mistry does in his lab, in our lab. So it's a confirmatory marker in addition to chitotriosidase, because some individuals actually have a mutation in the chitotriosidase gene that renders it inactive. So we want to make sure that actually we follow the right thing. So this is the cartoon of the gene, and all the nomenclature actually is the previous nomenclature. Now you need a 39, which is the targeting sequence, to read the new papers, okay? I am old, I'm keeping this because when we talk to the patients, we still talk about L44P, so on and so forth. So the gene is a bigger gene, and if you remember, there is an ethnic predilection, so it is more common in the Ashkenazi Jewish, and we look at it as actually N370S, the hallmark for type 1 disease, is claimed to be the most common mutation. However, actually the most common mutation is the L44P all over the world. Okay, it's a pan-ethnic mutation. It exists both in the Ashkenazi, non-Ashkenazi groups. And the homozygosity, so when you have two of these, actually it is an indicator of maybe there may be neuronopathic involvement. We're going to talk about that too, because the classification is based on clinical findings. So a molecular diagnosis may help, but a kid comes with a molecular diagnosis, completely normal maybe. Unfortunately, we need to wait and see where the baby is going to be, okay? So one thing about the gene is there is this hot spot between these 9 and 10 because the gene is a complex gene, meaning there is a pseudogene that goes in the opposite direction. So the pseudogene doesn't have any gene product, but what happens during conception is it messes up the actual gene, okay? So usually we see some very complicated genotypes that has a pseudogene sequence, which means the pseudogenes went into the gene itself and it interrupted the normal gene structure and function, okay? So I receive calls from frantic parents, so they say, now the things are doing a little bit different, okay? With the next generation sequencing, basically, it is not as looking at the gene one by one, but doing a panel of genes. So they may report actually 3 or 4 variants of GBA. Actually, the parents do not have 4 different mutations, but they have 1 allele, and the alleles are the pseudogene sequence that indicate a complex allele. And these complex alleles actually have been described by our friend and colleague, Dr. Nahid Taebi, who's working with Ellen forever, and she used different enzymes to cut the gene and then define the complicated genes with how these enzymes cut initially in the original experiments. So one thing with L44P is an interesting mutation, and the reason is I receive, actually now we have either the neuronopathic patients that are in their adulthood, they did well with the enzyme therapy, they're ready to have a family, or we have seen some babies that we have only found one parent to carry the L44P and the other parent, and the other parent's extended parent had no whatsoever at all. So there is only one mutation, which means that the L44P could get duplicated. Actually, we have cases that the chromosome on 1, where the gene is, got duplicated and inherited only one parent. So we have what we call uniparental disomy. We have 2 patients like that. We have one of them is a full chromosome duplication, and we have another 4 or 5. So the number is about 6, 7. We have kids with only one L44P inherited one patient, and the other one is a reason de novo. So this becomes really, really complicated when you do the genetic counseling, especially prenatal or preconception counseling, because it is not straightforward autosomal recessive, or you have Gaucher disease and your partner does not carry any gene, so all your children, hypothetically will be carriers, but they will not be affected. So I always say that this is biology, unfortunately it's not math, so there is a slim chance that you can have a child with Gaucher disease. This is very, very scary for the parents, but we have seen instances like that, so it happens, and we cannot give a full recurrence number too. And one more thing with the next generation sequencing is, so with these panel or the risk panels that are done through the labs, actually there are respectable labs may have missed some unusual big deletions or recombinant events. We had one patient recently, the mom was carrying the L44P, and they did send another NGS panel onto that. It was normal, code by code, or no variant has been found, and then behold, the baby is proved to be a neuronopathic Gaucher disease. So basically the NGS missed the actual variant. So what is really important is when you have a neuronopathic allele in one of the parent, then you have to have a full gene sequencing in a respectable and reliable lab. So this is just a very brief what Dr. Mistry so eloquently actually talked about for 40 minutes. So basically, I'm going to introduce another concept here. So the stored substrate or the substrate is very inflammatory in nature, especially lyso-Gl, GL1, GB1. And it causes immense cell death. So we are introducing another nomenclature, whatever is what is called danger-associated molecular patterns, or they're called DAMPs. And the DAMPs are everywhere. So basically, they're telling the cell to die because it is so dangerous to the other cellular structures in the environment, the cell cannot survive. So when we are looking at the neurologic phenotypes, there is intense cellular death due to the inflammation. So there is inflammasome activation due to this, either substrates are acting as DAMPs, or so there are other signals. And when actually you can control the cellular death, you can also decrease the substrate deposition or the macrophage activation. It becomes a vicious cycle. The cells die, macrophages get activated, and the other immune cells, and then immune cells activation causes cellular death. So after the COVID, we put our hands on a substance that is what's called a pan-caspase inhibitor, and we have some preliminary data. I didn't want to show it here because it's early. We have patents on that. That is basically to treat, the LSDs or Gaucher disease as adjunct therapies to control the inflammatory pattern, because even though you treat the patients, the inflammation still stays. I mean, you cannot bring it to zero. Probably the reason the events have started much earlier than the patient's diagnosis. So there is a point of no return that's related to inflammation and cell death. Similar to when we look at the brains is type 2 brains, or these are the babies that die. The brains actually are affected in utero, and there is much gliosis, which is actually akin to scarring in another tissue. So when you look at the Parkinsonism or Parkinson disease phenotype, it is the undigested material that is found in the cell. So this is the scope of the pathology of the neurologic involvement. So why there is a big spectrum of neurologic phenotypes? So we can have a baby with an anticipated GD2 phenotype in the future, whereas they don't turn out to be, okay, and we have patients such that. So the theory is, obviously, there is then association of the lowest actual enzyme levels are associated with the more severe phenotypes. Usually zero enzymatic activity is not compatible with life, and there is a threshold, which is about 10%, that is, that may maintain or make it to a milder phenotype. So for most lysosomal storage diseases, there are correlations have been observed between the residual enzyme activity and disease severity, and that holds for every single disorder that we see in our clinic. Whereas the null, whereas the zero, and very severe GBA1 pathogenic variants result in a very, very low GCase enzymatic activity. So this is given, okay? So when you have null, zero, or very severe, then you expect the patient to turn out not to have a great prognosis. But when it comes with a 2 missense mutations, so these are the letter change mutations. So these are not actually processed what is called the UPR, because they don't have or have very little protein product. They do have a protein product. If you can rescue them, you can also rescue the phenotype to a certain extent. So this is, as this says, this is not an absolute predictor of an outcome, especially for the missense variants. So L44P, that is linked to the neurological forms, the phenotypes associated with this is non-neuronopathic or very mildly neuronopathic. I have actually patients who are IT specialists, math professors, who have this genotype. They have very, very mild eye movement issue, and that's about it. And one of them had one seizure kept in with the seizure medication, but you need to really, really look for it to see the eye movement issues. And some L44P homozygous are very severe, actually turn to be type 2s. So one of the hypotheses is how the missense mutations are processed in the cell and whether some of them could be rescued can be a determinant of the phenotype. Why I'm talking about this, because we're going to talk about a little bit the chaperones, Ambrox also, and so forth. So this is the cartoon. Actually, this is from Ellen. So basically, these are the saccades, which is the you move your eyes right and left. And there are two nerves that comes from the back of the brain. There are 4 and 6, and 6 nerve has the longest route in the brain. It is the first one that is impacted in neuronopathic Gaucher disease. So there is a video online. The best way to do it, not to move the eyes right and left, but have the patient look right and left as you are moving your fingers, okay? Then what you'll see is you will, as an examiner, they tend to move their eyes by blinking, okay? So these kids actually blink a lot, not because they need to blink, but these are required to move their eyes, okay? In extreme, there are phases to it. So basically, the very mild case, you have a slow saccade with frequent blinking, and then when it's total paresis, this is paresis, which is like it's not working well. When it's plagia, which doesn't work at all, you have the squint. So basically, the eye will turn inward. And first, we see the patients, actually, it comes and goes, and it becomes fixed. And the first step is the doctor sees it, and the second step, the family would see it. So the family, one of them actually said that, oh, my daughter started turning her head in a different way. It just occurred like 7 months. Yeah, this is when actually the horizontal saccade slowing of paresis started. It's very actually obvious. And then it moves actually forward. You have the squint, you have the fixed squint, and then the vertical saccades are impacted too in the extreme cases. So when you see a very severely affected child or a baby, their eyes would not move. They're illiterate. So what are the other suggestive features of a neuronopathic involvement? This is almost universal. It's background slowing of the EEG, which indicates there is brain involvement or encephalopathy. So this is not, actually doesn't correlate with the clinical outcome, but it is a finding that shows that the brain is impacted. We have patients with this finding who graduated college, that was very successful, became lawyers, such and such. However, I need to attract attention to one thing, because we have usually an argument with the neurologist about the seizures, okay? So the family send me videos, the kid is seizing, and then the EEG is negative, is that right? And the neurologists don't want to do anything. Please don't do it. The reason is these are subcortical or EEG negative seizures, which means that the seizures start from not the structure in the cortex, but deep structures orthocerebellum. So they are indicative of more severe seizures. So we want to control them as much as we can, so that it doesn't become an emergency. You call the 911. Okay. So I treat all the seizures that are clinical with the seizure medications. So the other one is if you have a homozygous.
Can you talk into
Sure.
the mic? It's hard to
Yeah. So it is, the other thing is if you have two L44Ps, then it means that the patient will have a neuronopathic involvement, but we cannot determine whether it's going to be mild, severe, so on and so forth. We follow about a little bit less than a dozen of adult patients with L44P homozygote. Some of them are very impacted, and some of them are very mild. I mean, one of them just got married, manages a farm, and they are actually getting ready to have a baby. So one other thing is with the L44P homozygosity, and we see this not very often as European countries. There is progressive kyphoscoliosis or kyphosis, which is neurologic scoliosis, is from the bone. And so this is, they do occasionally bracing. Since this is neurological, the bracing may or may not work. And these patients, if the curvature is steep enough, they get actually fixed. And we want to fix them before it becomes an issue for both the breathing and also nerve entrapment. So the other thing is the hallmark is almost auditory involvement. So we do actually ABRs on these newborn screening babies, and if the ABR is abnormal, basically it will be a neurological patient. If the ABR is abnormal, doesn't mean that it's going to be type 2 or type 3, we wouldn't know that. But if the ABR is normal, then probably they're out of the woods, at least for the type 2. However, if the ABR is abnormal, it doesn't mean that they can't hear, they hear well. We have no issues, but ABR means that there is a brain stem dysfunction, basically, that involves the auditory system. But in the later years, they may develop hearing loss, and some of them may be abrupt. So it is actually very important to follow the hearing when they are the kids are verbal. Then we start doing the normal auditory evaluation. And then there are the issues with the severe disease, such as Gaucher, big lymph nodes, infiltrative lung disease. So this is the non-neuronopathic but systemic involvement-related issues in the neuronopathic Gaucher disease. There is a particular type. It is called type C. I have one patient that we follow of Eastern Asian origin. So this phenotype is associated with the heart involvement, the heart valves, and usually the demise actually happens because of the progressive valve involvement. So these are the extreme cases. You're looking at a baby with skin involvement now. And unfortunately, the babies with skin involvement, they are impacted in utero, and we come to understand that there is no salvage for those, and even the gene therapy is not going to be effective. Okay, so myoclonic epilepsy, encephalopathy, that is referred to type A. These patients may have less systemic involvement, but they may have a certain course that they may get wheelchair ridden, and myoclonus is very specific to the Gaucher disease and other LSDs. It is a very hard to treat epilepsy pattern. Usually these patients are 3-4 different anti-seizure medications, and as one of the moms said, unfortunately, myoclonus came with a vengeance. So basically, it impedes their mobility and what they want to do. So it is a very, very hard to treat type of epilepsy, or actually in this case is the brain involvement. And also in rare instances, psychiatric disease is noted. This is very common in the Egyptian population. We have 1 or 2 patients with severe psychiatric disease or psychosis that we think is the part of Gaucher disease. So even the type 2 has a spectrum. We thought that they were more ubiquitous presentation. So you see different presentations here with the brainstem involvement, skin involvement, to normally appearing babies and kids that turn to have severe course and unfortunate death by age 2 to 3 years of age. So the definition of GD2 is, I don't want to read the whole slide, but when we are not sure about the type, the caveat is if they walk, if they become ambulatory, they're not considered to be type 2 anymore. So this is walking by or before age 2 is the hallmark for lack of type 2, okay? Because type 2s with the current management can live up to 5, 6, whatever, farther than that. And if they don't attain independent gait, then you have a type 2 baby. So these are some of the type 2 babies. Actually, if you can anticipate this, the patient is on the left side, is very mild, and actually she was standing by herself. We have that kind of babies who seem to be almost normal, and they die in their sleep of central apnea. So we come to realize that it is very important actually to do sleep studies, and we start them on BiPAP or CPAP if they have central apnea. So these are the unmet medical needs, and the most unmet medical need, obviously, babies die. And so with the advent of gene therapy, you're going to hear a lot this afternoon, I believe, from Prevail. We have evaluated and assessed babies, and then deemed some of them actually to be proper for gene therapy before the onset of symptoms. Okay, so this is the important part. We want to send the babies before they are clinically impacted, but the caveat is how to understand that. I'm going to talk about a little bit. We do some different things in our lab and in our clinic, whom to refer and whom to still watch, because I have one child that was supposed to have a type 2 and she's walking, talking, and has some slow saccades, but that's about it. So this is the type 3 before the advent of ERT. It is very heterogeneous, and we talked about that, but let's talk about this L44P. This was 32 patients that we wrote with Ellen together at the NIH. So basically, it's a whole spectrum, and then we discuss some of the reasons why we see this spectrum. Obviously, this is not something we wholly understand, and it is very interesting. It's not contingent upon your enzyme levels. Some of them do well, and some of them don't. So these are some of the unmet medical needs. Even though we can treat systemically with enzyme therapy, we still have, because the disease was so severe, the systemic involvement. So I will give an example. Okay, so we talked about lyso-Gb1, is that right? It's the inflammatory substrate that circulates, and the origin is from larger organs. Probably it's coming from either from exocytosis or the disruption of the lysosome and the cells. So we did a not an experiment, but it was actually one mother. So we did the lyso levels before the infusion, right after the infusion, and a week after the infusion. So I'm going to give some arbitrary numbers. So before the infusion, it was 90. After the infusion, it was 45. And 1 week after, it was 75. These are some real numbers. So which means that the disease is so severe, basically you can never have your lyso levels normalized or even acceptable levels, that is for treatment, which I would like to have less than 20, while these patients are on treatment. Second is, the disease is so severe, it collects, the substrate collects, or you can measure in the periphery a week after to untreated or right before the treatment level. What I'm trying to say is, basically all the other disorders of infantile presentation, like MPS, and we see quite a bit of MPS patients now. So they do weekly infusions. This is FDA approved. The only one therapy for the LSTs that is not approved weekly for the infantile presentations is the Gaucher disease, because mainly the trials have been done on type 1 and they were grandfathered to type 3. So basically, every other week may not be the appropriate infusion frequency for a type 3 or a more severe patient with Gaucher disease. That's what I'm trying to say here. So I'm going to talk about just very few because Chris but kind of brought up this. So newborn screening and Gaucher disease. This is a kind of debate. I think, Aviva, you actually sent a RASP request. And what is RASP is Recommended Uniform Screening Panel. And there are 35 disorders in RASP. And Pompe disease is included in RASP. And there are some adjuncts, secondary 26 other disorders. So what is to be included in RASP? You need to have an infantile presentation that Gaucher disease has. There needs to be markers that is effective, that goes with the treatment, that goes down with that there is. And there is a reliable test, which there is. But the third one is there needs to be an effective treatment, which there is not. Okay. So Gaucher has not been in the RASP for the reasons that is the most important here is there is not an effective treatment for the infantile forms that we see right now. So whether gene therapy will offer an effective treatment, I think we'll see, time will see. And so what we are doing for these type 2 or the babies that are diagnosed through newborn screening. So we're following 7 of those babies from the day that they were diagnosed, and then we try to figure which one will be type 2, obviously with the molecular diagnosis, but with the clinical presentation, so we can refer them whether gene therapy would be an effective measure. So one thing is very important, as Dr. Mistry said, actually systemic inflammation or systemic disease also impacts the brain, okay? And the systemic disease will forward the brain disease independently, which means that if you have a severe systemic disease, this will actually contribute to brain degeneration independent of the brain degeneration. So we have seen this, and this has been done in Egypt, that they have seen these very severely impacted children during the shortage. Their neurological disease became worse because their systemic manifestations were not treated. So one thing, obviously, ethically, before the gene therapy trials existed, is to counsel and refer the type 2 patients to hospice care or end of life. So when I see those patients, I will not do that because we really don't know which one will have hope and which will not. So basically, we treat them aggressively so we can keep them healthy systemically as much as we can so that we can prevent some further brain impact with the prevention of the systemic disease. So we accept any patient. We don't care about the insurance. We have some donated, actually, enzyme therapy samples. So if anybody has some unused or wants to donate, we can put them in good use, and we do not charge these patients, and we really don't care what insurance they have. We just want to have them and have them treated ASAP. So let's talk about the current landscape. So basically we have the ERT and the SRT and the in vivo gene therapy. I'm just going to touch base what is being done at LDRTC. So Dr. Mistry a little bit described about the Vanglustat study. So we inherited the Vanglustat study from Baylor, Rafi Shivman during the pandemic because we worked through. So we follow those patients. They switch from enzyme plus SRT to mono-base now, and Lauren is the coordinator for that study. Obviously, we're not at ease to share those results, but these are some of the published. So basically, Vanglustat, which is a glucosylceramide synthase inhibitor, but the difference between eriglustat or Serdalga, it can cross actually the blood-brain barrier. So the first phase of this trial that we follow those patients, they did well. There were no safety concerns, and also actually that with the CSF measurements, they proven that the Vanglustat actually can access the brain. So these are some of the baselines. So basically the biomarker levels came down in that the first phase of the study. So we are gathering information on the long-term mono phase of this original study. We are also started actually as the first site in the world doing the mono trial, which is the lead mono, that the patients, adults and children, we have 5 patients in that trial, 1 adult and 4 children, is that right, something like that? That we are treating with only one treatment modality. So it's blinded, either enzyme or SRT, Vanglustat, and then at the open phase that they're all going to be treated by Vanglustat. So this is such a new study, and we recruited a lot. They told us not to recruit. I said, I have a lot of patients, we can contribute, but I guess they're going to have the other sites recruit more patients for that. So a little bit about the chaperone. This is the white elephant in the room, okay? The reason is, we talk about ambroxol, is that right? This is actually Mucinex is the derivative, the parent derivative of ambroxol, and ambroxol exists everywhere under the sun except for US. So I think in US it was never approved, and then after it was found to be a chaperone, there was a patent issue. It's a long story. Anyhow, so it doesn't exist as a therapeutic agent in US, period. And so I will tell what's happening with ambroxol, but what we have done with ambroxol, actually, we can test the blood cells, what we call PBMCs, and also the skin cells, whether the patients would respond to chaperones or not, because it is truly personalized treatment modality. So if you look at 3 siblings, one of them would respond. Obviously, they have the same mutation and a lot of the genetic environment, and the rest do not. So what we do by testing is, obviously it doesn't exist, and I will actually divert them to other physicians in the world that had used ambroxol, basically Japanese and Korean groups. But we can also give some sort of prognostication with using ambroxol from the hypothesis that if you can rescue some of the protein product, then they may have an attenuated phenotype, not as severe as it's expected, okay? So actually the responding and non-responding can be day and night. So obviously it is also dependent when you start, how much the patient is impacted, so on and so forth. But in our experience, the ones who do not respond to ambroxol in a petri dish, they tend to clinically do not well. So we actually send them to gene therapy without even, considering to observe, okay? Even if the molecular genotype is consistent with a neurological phenotype. And also ambroxol has other properties. In addition to being a chaperone, actually ambroxol has been used quite often now in the COVID pandemic. because it is an anti-inflammatory. And how it exerts its anti-inflammatory properties is it wasn't understood. There were many like T cells, so on and so forth, but we found that ambroxol actually can act as a what's called a caspase inhibitor, so this is also prevents the cellular death. So it is not as other pan-caspase inhibitor is not as robust, but it's one of the properties of ambroxol in addition to what is happening. Actually, now we have an IND, which is called Investigational New Drug Application to the FDA, that we are bringing ambroxol. It's almost done. The end of this month, thank you for after 1.5 years, the minimum that was actually required to be purchased was 1 million doses, so it did happen. So I have an IND for another disorder that actually it's in the clinicaltrials.gov. The first phase after that disorder has been reviewed, then hopefully we're gonna have Gaucher patients go through that. So the gene transfer, you're gonna hear about a lot this afternoon. I'm not gonna go about this. The last thing is ERT penetrant. Actually, it's brain penetrant ERTs. This doesn't exist for Gaucher disease, but there is a slew of studies, preclinical and early clinical, that is gonna happen at some point. There is one brain penetrant ERT has been actually approved in Japan for MPS II. They are actually getting approved in Brazil, and it's getting ready to be approved in US. So this is the second step is gonna be Gaucher is one of these disorders. So basically this concludes my talk, and I thank to everyone, but especially the parents and the children who let us be touched with their lives. Thank you.
Thank you so much to our three superstar doctors, Dr. Goker-Alpan, Dr. Mistry, and Dr. Sidransky. I mean, as Cindy mentioned, we wouldn't be here without them and without their entire teams. Unfortunately, we don't have time right now for questions, but they're going to be with us for the entire day, I believe. And so, when we're out in the hall during lunch, please, catch up to them and ask them your questions. And so, yeah.
So the Takeda presentation is shorter than we expected, so
Oh.
we have about at least 15 minutes for some Q&A.
Oh, great. Okay, great. So yeah, so if you guys want to, we have 15 minutes. Perfect. That's great. Yeah, for you and me.
I start, so I'm gonna...
Okay. Thank you.
On the road.
What is that?
We've got a good repertoire here.
So I had a couple of questions for Dr. Sidransky on the Parkinson study. So the first question that I had was, did you find that it was a particular Gaucher's gene, like one specific one, or was it any of the particular Gaucher's genes that led to an increased risk?
So all of the different mutations are associated with an increased risk. Even a few that don't even cause Gaucher disease. There is some feeling in the Parkinson community that the type of mutation might affect the risk and how severe the Parkinson disease is, but the jury's not totally out on all that. But definitely it can be any of the mutations.
Okay, and then the other question I had in terms of Gaucher's patients who develop Parkinson's, were the Gaucher's patients who develop Parkinson's, were they treated Gaucher patients? And do you think that that makes any kind of difference?
I've seen it in treated and I've seen it in non-treated. Sometimes even in New York for a while, the most common presentation for new Gaucher diagnoses was Parkinsonian manifestations, and then they'd sequence them and find out that they had Gaucher disease. So that would be in the group that's non-treated, and then many of them that reach our attention that have been treated for a while.
Hi, thank you all for your work, and I feel like your family sitting up there, known you all for so long. So I want to talk about I always read articles about irregular sleeping and that leading to dementia, and I know my brother and I both have really bad sleep patterns. Richard landed up with Parkinson's. Should we be, if we don't sleep, should we take sleep aids? Would that help? Is that good or bad?
So one of the prodromal things that sometimes is seen with Parkinsonism is something called the REM sleep disorder, and it's fairly specialized, and you can do a sleep study and find out if you have it. And those are people that tend to act out in their sleep, like hit people, and they're active in their sleep. Other than that, I'm not sure that we can focus on, we like all groups of people, there's some people that sleep better and some that don't. I think good sleep is good for all of our health. It's probably helps fight inflammation and other things. Sleeping is good for the brain, but I don't think that sleep aids or something of that sort is necessarily the way to go. I think that probably you should consult with a sleep specialist and see, let them do a sleep study and see what they see.
Any other questions? Well, you have them?
Hi, this is Wayne. Which disease inoculations should persons with Gaucher disease get? For example, influenza, pneumococcal pneumonia, meningitis, COVID. What else can we protect ourselves from?
Well, you definitely should get all of the normal vaccines that are age appropriate for you. If you're the age group that should get shingles and should get the larger dose of flu vaccine, I think that that's all appropriate. All the COVID vaccines are appropriate for patients with Gaucher disease. If your spleen has been taken out, you should have received a pneumococcal vaccine. What other vaccines do we need to bring up?
Pneumococcal and shingles and what else? Obviously flu.
There's no real contraindication that I'm aware of to any of the age-appropriate vaccines from early childhood all the way to late adulthood. Go ahead.
I'd like to make a comment about Ted's question about sleeping. And as Dr. Sidransky mentioned, the REM sleep movement disorder is very specific for Parkinson's disease. But it's important to keep in mind that families that are affected by devastating Parkinson's disease, as Ted is, unfortunately, that anything like sleep disorder that comes up, it can fill the mind with terror. That is this the beginning? So it is important to get a proper diagnosis, I think, before jumping to conclusions, because the level of anxiety in these patients are very high. But sleep disorders should be taken seriously and be evaluated. Even slim individuals, like most of us here, don't actually suffer from obstructive sleep dyspnea. A lot of people think that obstructive sleep apnea occurs only in people who tend to be a little overweight. But you can have obstructive sleep apnea in people with even low body mass index. So a formal evaluation can be very helpful. And there is now very credible, high-end clinical evidence that sleep deprivation can promote neuroinflammation and inflammation in the body. And so I come back to the agenda, the topics that are covered in the Gaucher Community Alliance meeting this time. And I do think that, I've come to these meetings for a long, long time, and our focus from physician community has been primarily on biomarkers, genetics, liver volume, spleen volume, different types of Gaucher disease, and so on. I think I see Gaucher disease in the 21st century as holistic care. And so, for example, practically, if you think about sleep disorder, and it turns out that you don't have obstructive sleep apnea, you don't have REM sleep disorder, then to be guided by the expert on what is the best agent. So a lot of people take lorazepam, it can actually affect memory and so on. So you need to really be guided by a professional and extend your multidisciplinary care team around you for each of the problems that you are experiencing.
And about the vaccines, actually, we encourage all the individuals, children, and adults to receive the vaccines. And then I get this question about the immune issues that Dr. Mistry had a very detailed explanation. So what we see is immune dysfunction with immune activation. But when we look at the vaccine response that we did actually uniformly for pneumococcus, we actually vaccinated the adults and kids and measured antibodies. They were all very appropriate vaccine response. So we encourage everyone to take the vaccine. If you have issues or concerns, we can always measure the levels or do that kind of trial.
Hi, I am wondering, in terms of research that's been done recently on types of treatment for type 1 Gaucher, if there's anything that's come out about symptom-specific help, like if there is a certain treatment that's good for certain symptoms specifically, if anything has been found in that field recently?
I'd be happy to take that. And so, the approach in the clinic is generally to treat all of the patient. And really, something that I train my mentees and fellows and residents is to really listen to the patient. Because even simple symptoms like aches and pains or not having low energy can have a real basis to it. And so one can do high-end testing, which I'm sure you've had, MRI, bone density, biomarker, and then all of that has to be put in a context. So the first priority is definitely to manage Gaucher disease, to suppress inflammation. And as I discussed, inflammation has many different phases, Even a very interesting comment from Dr. Alpan was that they measured the level of this inflammatory lipid before infusion and after infusion, right? And it fell and then it went up right then. That's telling you that you are dealing with a condition that is very dynamic, a bit like a mood disorder, right? That it is going to change and fluctuate based on your treatment and so many other things, if you have flu or COVID and so on. So I think it's very important to focus on that. The other thing that we have learned, and I tell my residents, the fact that you have Gaucher disease doesn't mean that you can't have something else. A lot of things that happen to you can be traced to Gaucher, but there are some patients who have two different disorders at once. Genetic disorders, surprising, because Gaucher is rare. How can you be so unlucky that you have a second disorder? But we have had a number of examples of that. And the most important thing is, even in those individuals, we can truly transform their health outlook and their lives. So I think it's a combination of listening, holistic care, and looking at not just the scientific data, but also symptoms, like what is your quality of life? That probably has a biological basis, and it's our job with all this testing to figure it out, right?
About the research, so our actual center does quite a bit of, we want to find biomarkers, and we want to, with the help of these biomarkers, we want to devise treatments. So we started looking into the bone density issues in patients with Gaucher disease. We think is primarily inflammatory, but there are different inflammatory involvement and then inflammatory there are treatments that are targeted to different molecules in that pathway. So we published actually 2 papers on that. Hopefully we're gonna couple with some treatment options, which there are more than 4 or 5 right now for osteoporosis, with the findings that we are, publishing. And I believe that there is not going to be one single uniform treatment for each patient. As Dr. Mistry said, they are the genetic factors, osteoporosis. Some families have run in their families, in addition to the Gaucher disease that actually brings as a risk factor.
Yeah, the other thing I was going to mention is that sometimes things that seem very simple can make a big difference. For example, a lot of our patients historically who've had bone problems and hip problems end up with a leg length discrepancy. And something as simple as putting in a little lift in one heel of a shoe can sort of change so much in terms of subsequent aches and pains and backaches and things like that. Sometimes something like an antidepressant can change somebody's whole outlook on how their disease burden is. So, it isn't always the big deal drugs. It sometimes can be other solutions. So it's important to find a physician that listens to you and they can work with you to find, what they can do to help the symptoms that are bothering you.
I'm going to have a question. So for patients with type 1 that everything seems well managed, your diagnostic tests and your biomarkers, like everything that way looks like this is a perfectly managed patient, but you have inflammatory markers that are way elevated, and I know that this affects a number of patients. Is it your opinion that there's something else going on other than Gaucher disease? Or is it still, even though Gaucher disease appears well managed in the diagnostic test, the inflammatory markers could still be related to Gaucher disease?
I'll start. I'll give the word to you since you have the most. Yes and no. So if you have a condition, we think it is the condition. However, inflammatory research and inflammatory markers are very hard to do a research with. I'll give you an example. We're trying to find controls. We can't. The reason is if you have a tooth infection, if you have influenza, the markers will go up and down. So there are a few things that we follow, which are the end of the pathway. One is the thrombotic pathway, and the other one is the B-cell markers, which we think is related to Gaucher, but we have seen other disorders, like COVID is an example, which the inflammation goes haywire and the patients succumb to it. As opposed to Gaucher disease, there is a smoldering inflammation that is there that the body can handle. What to do about this? Obviously, you treat the disorder, but there are, in my opinion, obviously humble opinion, we may use some adjunct therapies that we have not tried yet. But obviously, there needs to be trials for that, what happens to the disorder if we treat the disorder and something in addition to that.
So, Aviva, I have a good understanding of what your question refers to. This can be a very, very tough question to really answer in a categorical way. And to some extent, that reflects our limitation in how well we understand Gaucher disease. And so when you have your biomarkers done, that is at one moment in time. And so most people here have your Kito, lyso-GL1 maybe done once a year, twice a year, and that is one particular day. It doesn't tell you what is happening in a dynamic manner over a, let us say, 3-month period, right? So in people who suffer from diabetes, they have hemoglobin A1C. That's a marker that gives you 3 months of integrated blood sugar control value. And anyone who is with diabetes, they know their hemoglobin A1C number. So one limitations I feel in Gaucher disease is just this limitation of the biomarker, that even if we took one person, let us say, over 7 days, and just measured their biomarker levels every day, I'm sure they're going to fluctuate. And then the question arises, what is the best treatment for that? Do we go with enzyme replacement therapy given once a week, once a month, or once every 2 weeks, or go on something that works continuously in terms of substrate reduction therapy? The other level of looking at this is ultimately this inflammation that is happening in Gaucher disease affects the entire gene transcription program in the immune cells. And I think that the next stage of investigation is going to be to looking, just as we did with the microglia immune profiling, and we have begun that work to take blood, when we take the blood sample to isolate what's called PBMCs and do single-cell RNA sequencing to see, let us say, if we can identify an elevated level of interferon signaling genes, what is being indicated in our preliminary studies. So those are the limitations in terms of Gaucher disease, but at the next level is, again, it does boil down to, is there a second disorder? And like, let us say, inflammatory bowel disease or some kind of inflammatory arthritis. I have had people with two totally rare disorders colliding together. And that managing both of them can improve health prospects. So there are different levels and there are significant limitations we have.
Okay, this is going to be our last question. One more.
Hi, good morning, everyone. Thank you for coming. So this question is as a type 1 Gaucher patient, but also as a clinical psychologist that works with a lot of patients with medical conditions. I'm always encouraging them to try to understand the biological bases of their mood disorders. And so recently in a Gaucher chat room, somebody asked if there was a biological basis for mood disorders in Gaucher disease. And I'm really curious to know what all of your thoughts are about that. I don't know if there's an inflammation connection, for example, or some sort of biological basis that's been found for mood disorders or anxiety, any type of mental health issues with Gaucher disease. Thank you. Well, I think I stumped the panel.
I don't know that I can say that there's I think that patients that have chronic illness and chronic pain have reasons to have anxiety, mood disorders, depression, things like that. And so I don't know how to divorce that from whether that's physiologic or it's situational.
Yeah. So, as you can judge from my comments today, I've become such a big fan of this idea that people used to think this is hocus pocus medicine, right? And so I think neuropsychologists should be part of multidisciplinary team. Why shouldn't we think about getting specific patients to have proper neuropsychological battery of tests? In type 3 study, for example, we are using Arbenz testing, right? And so when neuropsychologists make an evaluation, I am always awed by the scope of functioning parameters that they capture. And I think we have a lot of important work to do in that area for healing whole of the patient and not just biomarkers or checking liver volumes and so on. So you mentioned you are an accomplished neuropsychologist, and I think clinical side. But forgive me, it's my ignorance, but I was referring more to neuropsychological battery of it. We tend to think about this test only in people in whom we want to make a diagnosis of mild cognitive disorder, but the whole battery of tests actually really captures the whole of the functioning of the individual. And in 21st century, I think we have to be armed with that data just as we are armed with the biomarkers or liver volume measurements.
So if the question is if we see any lesions in the brain that would correspond or explain the anxiety or the universal, depression, probably not. But having said that, this is akin to PTSD. So it is medical PTSD, and even like we try to normalize what is happening to little kids, this is not a normal situation, and they get traumatized, they get poked, they have MRIs, they need to lay in the machines, they miss school, and it becomes part of life. And you try not to be a nuisance to you, but it is what it is. I mean, you take somebody home, there's a stranger, you need to adjust your infusion. I mean, there are so many things that you need to be dealing with, even the sibling relationship, the spousal relationship. So I take it as a PTSD, and there is a treatment, and the soldiers that go to war, so we are having a war against Gaucher disease at different levels here. And the patient gets impacted